So first of all, I would like to thank the invitation by the committee of the Co-Partnership Forum. And today I'm going to share with you our recent work and mostly focus on the study of mosquito host factors that regulate dengue virus replication and transmission. As we all know that the emerging and re-emerging mosquito-borne disease remains a great threat to global health, including this disease, including Zika, West Nile, chikungunya, and dengue. And these diseases are widely distributed in tropical and subtropical region, and that resulted in about 390 million cases per year. And currently, the control for mosquito-borne disease, in particular dengue and Zika, is focused on three approaches. So first is to develop a vaccine. And although there's now a dengue vaccine in the market, however, the safety of this vaccine remains a huge concern. And second approach is to develop anti-dengue drug or anti-Zika drug. But and then we know actually that there are some medication is under investigation, but still not available in the market. So currently the control of mosquito-borne disease is mainly relies on the vector control. And most of the local government make use of spreading of insecticide as the control approach. However, the overuse of insecticide will increase the resistance of mosquito to insecticide. So to overcome this problem, several alternative approaches such as transgenic mosquito or biological control, for example, Wolbachia-based approach has been developed. So to support these novel approaches, the thorough understanding of mosquito virus interactions is still essential. So our study is mostly focused on this issue, the mosquito virus interactions. So when a mosquito taking an infectious blood meal, the virus replicates in the mosquito midgut. And next, the virus disseminates to other tissue, for example, the cervix gland. And once the infected mosquito take another blood meal, the dengue virus is then released from the cervix gland through the saliva into the host. So during the virus dissemination in the mosquito and transmission to a new mammalian host, that dengue virus actually interacts with a variety of mosquito proteins, which are very unique and is also important during each phase of the virus cycle. So in our previous study that we make use of a hemorrhage mouse model by using a step 1 knockout mice. And then we have found that when these mice, if they are infected, if they are intradermally inoculated of dengue virus, and then those viruses are produced from a mosquito cell line 6636. And then these mice will develop a slight hemorrhage sign in the back skin and abdominal skin, as you can see in the upper panel. However, if these mice are intradermally inoculated with saliva from infected mosquito, that will develop a severe hemorrhage in the back skin and abdominal skin, as you can see in the lower panel. So we speculated that mosquito factors play important roles in modulating dengue virus-induced hemorrhage development in the mice. So we next make use of NGS analysis to examine the transcriptional regulation of gene cohorts that are in response to dengue virus infection in the mosquito Aedes aegypti. So we select mosquito midgut and cervix gland as two target tissues because these two places are the location where the dengue virus replicates. So at the end, we found 7 genes are upregulated in both midgut and salivary gland. So we select 3 candidate genes for our further analysis. So we infect the mosquitoes with dengue virus and examine the transcriptional expression of apirus and D7L2 and AASG34. And from our result, from our qPCR results, that we have shown that these 3 genes are found to be specifically expressed in the salivary gland after infections. And then we therefore examine the effect of these 3 genes on dengue virus replication. So by silencing each gene through the RNA approach, so we're silencing this gene before dengue virus infections, and then we found that dengue virus E protein production in the mosquito salivary gland was affected. So our result shows that when silencing AASG34, we can see that by Western blot analysis, the dengue virus E protein production is significantly inhibited in the mosquito salivary gland. And lower panel is the quantitative data. So this data suggests that adisogyptide AASG34 is critical for dengue virus replication. So we next examine the effect of ASG34 on viral protein production in the mosquito salivary gland and saliva. And here we show that when silencing ASG34 in the salivary gland, that can completely inhibit the release of dengue virus into saliva. Here we can see that at day 7, day 10, and 13, that we can barely detect any signal of E protein production. Here we have shown that by a Western blot analysis, but as well as the plug assay that we can see that when silencing ASG34, that we can see that the infectious virus particle production is almost inhibited in 7 and 10 and 13 days post-infections. So this finding suggests that silencing of AASG34 not only inhibit dengue virus replication in the salivary gland, but also prevent the virus release into the saliva. So we want to investigate the effect of AASG34 on dengue virus transmission. So again, we're using this mouse model, the state 1 artificial mice. And then this mice was first infected with saliva that were collected from control mosquito, the laxative-treated mosquitoes, or infected by with saliva from AASG34 silencing mosquitoes. And then both mosquitoes are infected with dengue virus. And then we examine the development of the hemorrhage at day 6 post-infection. So here we can see that a severe hemorrhage developed in both dorsal and abdominal skin in the mice that are inoculated with saliva from infected control mosquito. However, in the mice that are inoculated with saliva from infected AASG34 silencing mosquito did not develop a hemorrhage in both skin regions. And also we look at the spleen. So here we can see that the spleen of the mice that are inoculated with saliva from infected control mosquito that are enlarged. But those of the mice that were inoculated with the saliva from infected AASG34 silencing mosquito remain small. It's around 70 milligram. So this result indicated that the mice shows an active immune response to the saliva from the infected control mosquito, but almost has no immune response to the saliva from the infected AASG34 silencing mosquito. So this data suggests that silencing of ASG34 inhibit the transmission of dengue virus from the mosquito to the mammalian host. So here I shortly summarize some observation from this study. So here we have shown that this cervical gland protein AASG34 is highly expressed in the cervical gland post-dengue virus infections. And then we have shown that this protein is essential for dengue virus E protein production in the cervical gland and the release from cervical gland to saliva. And then at the end, we demonstrated that this cervical protein ASG34 is critical for dengue virus transmission. And this data has been published in 2019. But however, there's still questions related to mosquito virus interaction. For example, whether the mosquito vector that can change the virus that resulted into the consequential disease manifestations. So to answer this question, again, we use this dengue hemorrhage mouse model to investigate whether the infection of mosquitoes will resulted any change in dengue virus, and then that will affect the ability of the virus to induce hemorrhage development in the mammalian host. So we have 4 different groups. And the first group is that these mice are infected with dengue virus grown in cell culture. So we call that culture DEN2. And then the second group, we have mice infected with this culture DEN2 virus premixed with saliva from naive mosquito. So we call that culture DEN2 plus naive saliva. And then the third group is the mice infected with saliva from DEN2 infected mosquitoes. So we call that infectious saliva. And then the last group is the mice infected with culture DEN2 premixed with UV treated infectious saliva. So here we can see that in 4 groups that we can see the hemorrhage development in dorsal skin and abdominal skin in all groups of mice on day 6 after infection. But it is important to note that mice, if the mice given an infectious saliva, that will develop more severe hemorrhage with greater area of involvement in the abdominal skin than other group of the mice. So this result suggests that dengue virus in infectious mosquito saliva has higher hemorrhage induction potential than virus obtained from cell culture. So we use another experiment, so we further explored the involvement of the mosquito saliva in potentiating dengue virus hemorrhage inducing potential. So here we infect those infectious mosquito saliva was placed on the mosquito cell culture, and then the supernatant of the cell line was collected at 7 days post-infections, and those viral titer of the supernatant was determined by a plug assay. And then this virus obtained from one cell culture passage, we call that MSQ DEN2 P1, so which means that the P1 passage virus was again compared to culture DEN2 for their potential to induce hemorrhage in the stage 1 knockout mice. So again, we can see that when mice receiving this MSQ DEN2 P1, which means the P1 passage virus, that can develop more severe hemorrhage in the abdominal skin for those compared to those given a culture DEN2. So this result, together with the previous data, suggests that dengue virus infected mosquitoes that can acquire greater potential to induce hemorrhage in the mice. And even more, that we have from this data, we suggest that this MSQ DEN2 virus retained its hemorrhage potential ability even after one passage in the mosquito cell line. So we want to know that what happens to the virus when this virus has a passage into the mosquito body. So we compare the morphology of the MSQ dengue P1 virus, which is a P1 passage virus variant, to that of the culture dengue virus by TEM analysis. So we can see here the diameter of the MSQ dengue P1 virus particle is smaller than the culture dengue virus, but there is no difference in the shape of the variant between the two. However, the plaque size of the MSQ dengue virus, they are larger than that of the culture dengue virus. And also, we have using the plaque assay to examine the growth rate of this MSQ dengue P1 virus in 3 different cell lines. For example, in BHK, this mammalian cell line and 2 mosquito cell lines. Here we can see that this MSQ dengue P1 virus, they have higher titer than the culture dengue virus in all 3 cell lines, and they have a greater growth rate. So this result indicates that the infected mosquito that change the growth characteristics of dengue virus, and this change is shown in its particle size and plaque size and the growth rate, which they combined, they can affect the greater, they have the effect in a greater hemorrhage, inducing a potential in the mammalian host. And we compare the MSQ dengue P1 to culture dengue for their ability to induce macrophage TNF production. So here we're using a qPCR to analyze the TNF production. And then here we can see that the MSQ dengue P1, this virus that induce almost 4-fold higher TNF production than culture dengue virus, even as early as 24 hours after stimulation. So this data indicating that MSQ dengue P1 is more efficient than culture dengue virus in triggering TNF production. So this data also demonstrate that there's a correlation between the efficiency of MSQ dengue P1 in inducing TNF production, and also shows that there's a enhance the ability to potentiate the hemorrhage development. So we speculated that the infecting mosquitoes change the virus, that it became more efficient in triggering TNF. Therefore, they have the greater potential to inducing a hemorrhage in the mice. So finally, we concluded that dengue virus collected in infected mosquito saliva induce more severe hemorrhage in the mice. And then we have shown that dengue virus collected in infected mosquito, they have a smaller particle size, and then they have larger plaque size, and they have more rapid growth rate. And so we have shown that the mosquito provides an excellent environment that can change dengue virus by changing its growth characteristics as well as its potential to cause disease. And this work has been published in 2021 in PLOS NTD. So finally, I would like to, this work was supported by the Ministry of Science and Technology Taiwan, and also we have a lot of collaborators and students that has been working on this project. So thank you very much.